Hey everyone, welcome to our basic genetics problem solving tutorial. After watching this tutorial, you should be able to solve monohybrid genetics problems for traits that show complete dominance, incomplete dominance, codominance, and sex-linked inheritance patterns. Before completing this tutorial, you should have a good idea about DNA and gene expression, how sexual reproduction works, including meiosis, genetics terminology, and a general concept of Punnett squares. If you need to, go ahead and review this background material before getting started on the tutorial.
All right, let's dig in.
Our first example is one of complete dominance. We're going to look at fur texture in dogs. There is either wiry fur or smooth fur, and wiry fur is dominant over smooth fur. Before we get started solving the problem, which is down here, we are first going to need to look at deciding our notation. In genetics, we use letters to signify the alleles, and when we have a completely dominant allele, we use an uppercase letter that represents that. So in this case, a W would be appropriate to represent wire hair or wire fur, whereas we use a lowercase letter to represent the recessive version of that same gene. Further, we could say that a big W and a big W little w dog, both of those would have wiry fur. And the only time you would get a dog that had smooth fur is if they were homozygous recessive and had two copies of the recessive allele. Now that we have that basic information, we can go ahead and work on our genetics problem. So it says that we are going to take two wire-furred heterozygous dogs and mate them. And then we want to know what the expected genotypic and phenotypic ratios are for the puppies. The first thing we do is write out our cross. So we have two wire-furred dogs, so they're each going to have a big W. They're both heterozygous, so they also have a little w. So we're going to cross two big W, little w dogs. In order to use the Punnett square, which is down here, we need to remember that each of these dogs or each of these parents is going to produce gametes through meiosis and separate out these two alleles that are on homologous chromosomes. So half the time gametes will carry a big W and half will carry a little w, and that goes for both parents in this case. We're then going to take these alleles and move them down onto the Punnett square. For one parent, we write it on the left side, for the other along the top, and then the offspring will be in the middle. These represent the gametes here on the side. And then on the inside here with the boxes, these represent the possible offspring. That would be the result of fertilization. Sometimes we might get a big W from each parent. Other times we get a big W from one parent and a little w from the other parent. That could work the other way as well. And then you could also get a little W from each parent. Notice that we're working with 4 boxes. So when we think about the ratios, we're going to look at how many out of those 4 have each genotype. First, we're going to write out the possible genotypes. So some could be big W, some could be big W, and some could be little w, little w. Out of these 4 boxes, 1 is big W, 2 are big W, and 1 is little w. So that tells us our genotypic ratios that are expected for the offspring. And then we need to do the same for the phenotypes. So the phenotypes could either be wire-furred or smooth. And out of these 4, these 3 here will have wiry fur, and just 1, the homozygous recessive, will have smooth fur. So our phenotypic ratio is wire to smooth 3:1. Next, we'll look at an example of incomplete dominance, and we're going to look at hair texture in humans. This is an autosomal, meaning not sex-linked trait, with curly hair being incompletely dominant to straight hair. For this, we're going to use a slightly different kind of notation. We're going to use a letter that represents the trait. I'm going to do H for hair, and then we're going to have the specific allele represented as a superscript.
We'll have H superscript C be curly and H superscript S be straight.
Those are the 2 different alleles. For the genotypes, an individual could be HCHC, HCHS, or HSHS. And because we're dealing with incomplete dominance, we have a bit of a difference. So for the ones that have 2 HC alleles, they will definitely have curly hair. The one with the 2 S alleles, they will have straight hair. And then These ones here that are heterozygous are actually going to have in between curly and straight, which would be wavy. Wavy individuals are HCHS. With this information, now we can work on our problem. Our problem states that we have a curly hair woman and she's having children with a wavy hair man. So let's first write out the genotypes for the two parents. She's curly hair, so she's going to be HCHC, and he has wavy hair, so he will be HCHS. Remember that it is not sex linked, so it doesn't matter which one is the male and which is the female for solving this problem. For the woman, all of her eggs will have an HC. That's the only possibility she has to pass on. But for the male, half the sperm will carry an HC and half will carry an HS. For the offspring, they could get a HC from both parents, or they could get a HC from mom and HS from dad. So now we again want to write out the expected genotypic and phenotypic ratios. So for genotypes, HCHC to HCHS. So notice there's 2 that are HCHC and 2 that are HCHS. We could say 2 to 2, but it's better to just say 1 to 1. And then we're going to do the same for the phenotypes. So for the phenotypes, these ones that have 2 of the HC alleles, they are going to have curly hair. And these ones that are HCHS will have wavy hair. And again, the ratio for those will be 1 to 1. Moving on, we're going to look at an example of codominance. Codominance is when neither allele overcomes the other one, and instead you see the effect of both of them. We're going to look at feather color in chicken. It's controlled by one gene with 2 codominant alleles. So the 2 alleles are black feathers or white feathers. Let's write that out first. We're going to say F for feathers, and then we could be either FB, and that would be the black allele, or we're going to be FW, and that will be the white allele. For the chickens, they could be either FB, FB, so have 2 copies of the black allele, and they will have black feathers entirely. Take this one down here. Or they could be FW, FW, have 2 copies of the white allele, and be completely white, like this one down here. Or they could be heterozygous, be FB, FW, and then they would be a combination of black and white that we would call speckled. Now that we have this information, we can use it to solve our genetics problem. Our problem states that a speckled hen is crossed with a speckled rooster. In this case, they both have the same genotype, and it is also not sex linked, so it doesn't matter which is male, which is female. They are both speckled, so they are both FB, FW. For both of them, half of their gamete will carry an FB, and half will carry an FW. And then for the offspring, there's a possibility they get the black allele from both parents, or they could get one black allele and one white allele from each parent, or they could get a white allele from each parent. We're going to write out the genotype and phenotype ratios again. So the possibilities for the genotype is FE, FB, or FB, FW, or FW, FW. And 1 out of the 4 squares is FB, FB, 2 are FB, FW, and 1 is FW. FW. We will also figure out the phenotypic ratio. Some will have all black feathers, some will be speckled, so they have both, and some will have all white feathers. These ones here, FBFB, will have black, so 1 out of 4 will have black, 2 out of 4 will be speckled, and 1 out of 4 will be white. Finally, our last example is a sex-linked example. When a trait is passed on in a sex-linked inheritance pattern, that means it is on the X chromosome and not on the Y chromosome. The notation that we use for a sex-linked gene is very important. We have to remember that the gene is on the X chromosome and not on the Y chromosome. So we always use the X and then a superscript to show the actual allele. We never have a Y with a superscript. In this case, we have male pattern baldness, and it's caused by a recessive sex-linked gene. That means that we're going to use an uppercase for not bald and a lowercase b for bald. What you don't want to do is say Y big B or Y little b. Because it's sex-linked, those do not exist. When we think of the possible phenotypes and the genotype, we want to think about males and females separately. Females are XX, right? So females could be X big B, X big B. They could be X big B, X little b, or they could be X little b, X little b. These ones here are all going to be not bald because the little b is recessive to the big B. And then just these ones here that have 2 copies will will be bald. For the males, there's fewer possibilities. Males could be either X big B Y or X little b Y. So remember, the Y chromosome does not have the allele or the gene on it. So these ones will be not bald, and X little b Y would be bald. So notice that we have these ones here for the females. We have ones that are called carriers. They're not actually showing the condition or the recessive trait, but they are carrying it. So females can be carriers, males cannot. We also see sex-linked traits more commonly in males because there is not this masking effect. Usually the recessive gene is less common in the population, and so it is unusual to have a female that has 2 copies of it. Not unheard of, but just less usual. Now that we have our notation decided, we can solve our problem. So imagine that a man without male pattern baldness. He's a man, so he's going to be XY. He does not have the baldness, so he's going to have a big B. He is X big BY, and he is having children with a woman who is a carrier. She has 2 X chromosomes. One's going to have the big B and one will have the little b. We'll fill in these other alleles here in a second, but of course, if it's going to be a male child, it's going to get a Y, going to get that from the dad. Dad up here, half of the sperm will carry X big B and half will carry a Y. And for the eggs, half will carry a big B and half will carry the little b. So these ones over here in this column will be the females. They will get an X from mom and an X from dad. And then these ones here in this next column with the Y are going to be males, and they will always get their X from mom. When we're dealing with a sex-linked trait, it's convenient to separate out the males and females from each other and to just report the females separately from the males. These ones here are the females. For the females, they can either be from these 2 parents, X big B, X big B, or they could be X big B, X little b. And now we're just looking at the females, so we're just going to look at these 2 boxes instead of all 4. So half will be X big B, X big B, half will be X big B, X little b. For the males, we're just going to look at these 2 boxes, and we see that the possibilities are X big B Y or X little b Y. And again, it's a 1-to-1 ratio. So those are the genotype frequency for the females and males separately. And now we will look at the phenotypes. For the females, the phenotype is all of them are going to be not bald, right? Because these ones are carriers. Not bald, we'll just say 100% would be a good way to say that. Or if you want to say not bald to carrier, that's okay too, be a fine way to put it, and that would be 1-to-1. For the males, they are either not bald or bald, and just like for the genotype, it's a 1-to-1 ratio. All right, that concludes our tutorial. Today we went over how to use Punnett squares to solve monohybrid crosses for complete dominance, incomplete dominance, codominance, and sex-linked genes. Thank you for joining me.